Thank you very much, Gyril, and I would really want to thank the organizers for this to let me come and present what we have done during the past 10 years, I would say, in this field. And I probably have the longest title in this seminar series, but I wanted to include all the small bits and pieces that we can present here. So I will talk about real-time label-free platforms for interaction monitoring of drugs and nanoparticles with various types of things. I'm a real advocate of this real-time label-free measurements because, I think real-time measurements are very important because if you look on our body in itself, it's a very dynamic system, and many times we do in vitro studies in static systems. And if you think about what can happen in the body, you can have brain activities takes place in milliseconds. We have cellular processes, uptake processes that takes up in minutes, and then we have cell division that takes place in hours. So it's very broad time scale, and therefore I always like to look into the fact that we can measure something in real time so that we can predict what happens. And label-free is quite obvious because if we label something, we can then modify some of our material properties. And so basically, I had a group together with Alex Bunker at the University of Helsinki at the moment called Pharmaceutical Biophysics Research Group. And these are our PhD students at the moment. It's a group that actually do computational modeling plus then experimental tools specifically for real-time label-free monitoring. And I'm the experimentalist and Alex is the modeling guy. But the most of the work that I present today has been done by previous students or PhD students. So I want to highlight that their contribution is very essential for the results I'm presenting today. So as I said, why do I do real-time measurement? I had already mentioned that it's our body is a very dynamic system, or any biological system is very dynamic. And if we actually look, what we usually do, we stimulate, for example, cells in the beginning, and then we read out after a few hours. And sometimes it's difficult to then understand what happened. So with real-time measurements, we actually can then access the route, how we actually end up in this position. And then by doing real-time measurement, we can also hopefully predict what will happen at later stages. And we don't need to measure that later stages. So I see it as a complementary tool for these very good techniques we are using today in vitro. And so then using these real-time measurements, we can then access more information to understand our systems better. And of course, this gives us, from my perspective, better understanding of how drugs absorb or how they interact with cells and are taken up by cells. The other part is this label-free measurements. What I'm focusing on is because I have recognized, or even literature has recognized, that there are drawbacks by using labeling agents. And one is, of course, when we chemically modify our sample, our drug, our particle, our cells for imaging, we can actually then modify the physical chemical properties or then cell behavior even with these labels. And this is a very old example, but I like this example because it's so descriptive that if you have, for example, a gold nanoparticle that has been coated with a fluorescent probe, and you look on the cell uptake of this, this is basically the red dots in this graph on the right-hand side. So we can see that the blue dots here are different surface function relations of gold nanoparticles and the cell uptake of those. So there is some cell uptake, but immediately when you have fluorescent probe, it suppresses completely the cell uptake. They try to overcome this also to coat with a secondary protective layer the fluorescent molecules, and there's still very much suppression in the cell uptake. So therefore, I like to focus on fluorescent or label-free measurement techniques so that we can study the native samples and cells intact in their environment. And of course, we have always the question, are we actually detaching the fluorescent probe when we do measurements, or are we tracking our material or particle? And the technique I'm using is called multiparametric surface plasma resonance technique. Now, many people that has been using surface plasma resonance, for example, a Biocore instrument or something else, things like, okay, but this is an old technique. But the advantage of the technique I'm be using is actually that it actually scans the whole SPR spectrum. So that's what we are seeing upper left corner here. Basically, we have a movable laser and photodetector, and at a certain angle, when our laser hits a metal surface, all the energy of the laser goes to exciting electrons on the metal surface here. This is the basic phenomena of plasma resonance, surface plasma resonance. But the difference with our SPR instrument to any other on the market is that we do the complete scan here, and we can get a complete SPR spectra. Normally, only in commercial instruments, otherwise, they have a fixed angle, and you can then only detect a very small area of this surface plasma resonance. And what we do later on, we modify the sensor surface, for example, with a receptor, and then we can flow in a ligand, and then we can measure the interaction of these ligands with the receptor in real time. And you can see here up the right corner that the surface plasma resonance peak is moving to the right, which means that we have an absorption of molecules to the surface, and that tells us the kind of kinetics of association to the surface or between ligand and receptor. And then when we flush it, we can also have a dissociation of it. And the small dotted box here is what commercial instruments does measure in with this kind of instrument, but we can measure the whole angular scan here, which we also call this instrument a wide angular scan SPR, which actually allows us to measure with very thick layers. Traditional SPR measures with layers that are in the range of 100 to 300 nanometers, but we have been measuring with living cells, which are micrometers in thickness, as I will show later on, as well as also biofilms, which are 3 to 4 micrometers, and we still can measure interactions with these. And this cannot be done with any other SPR type of instrument on the market. So what I'm going to present today is more how we can actually use this kind of approach or technique to actually determine the size of nanoparticles. We can also determine the concentration of nanoparticles in our solution, and how we can study actually the formation of protein corona in physiological conditions. So we can use temperatures that resemble body temperature. We can use 100% serum, and then look on what proteins have actually attached on the nanoparticles, and then we can try to correlate that with how they behave in vivo. And then I will also present some data on how we can study drug or nanoparticle cell interactions, look on the interaction kinetics, cell uptake efficacy, and then also with biofilms, how we can study this nanoparticle interaction. So just to show you that this is not a black box, but it actually is based on physical realities, I will just go through a little bit about the background theory, how we do this nanoparticle size measurement. Basically, what we measure in its simplicity with this SPR is the refractive index change on the surface here near the sensor. And that is quite kind of understandable concept, because all material have a refractive index. And if we have a baseline starting without anything on the surface, and then we inject something and something absorbed on the surface, we actually get a refractive index change because the material increases there at the interface, and that is actually in its simplicity what we measure. So if you want to have a take-home message of what we are measuring with SPR is changes in refractive index. And that we can actually correlate this change then theoretically by this kind of equation that we have the SPR response is a function of the refractive index change as a function of concentration of our analyte, the bulk concentration, surface concentration of our analyte, the thickness of the layer, and also the decay length, what we have of our sensing kind of layer that we call an evanescent wave caused by the surface plasmons. And a sensitivity factor which is dependent on the laser, the prism we are using, the flow channels, and so on, what we are using. But this is a physical reality that we can utilize for actually do some deeper analysis of the SPR response, not looking only on the responses or affinities. And this is just an example of what these parameters now describe. But this is actually can be utilized now if we use two different wavelengths for detection. So how this is done is that if you have this SPR spectra scanned with two different wavelengths, there is now 670 and 785 nanometers. And the dotted line is the baseline or where we start. And then when we inject something, this peak moves to the right. And if you do this at two different wavelengths, there is a change in both peaks. And then if we take the ratio of these two responses, then we get an equation system like this. It looks rather complicated, but it's actually rather simple after all, because we can actually calibrate or measure this sensitivity ratio at two different wavelengths by using a non-interacting sample, glycerol or xylitol or something else. But then we need to know the DNDC ratio at two different wavelengths. But this is often known for proteins or lipids in literature. So, and it's quite close to 1 oftentimes. And then what we need to know is the decay length of our sensing depth of, of our system, which is dependent on the laser wavelength we are using, and that we can also calculate. So the only unknown we have in this equation is the thickness of the layer. So utilizing this then is that we measure these responses at two wavelengths. We can calculate this kind of, we can call it a calibration curve. We measure these two responses at two different wavelengths. And if the ratio of these responses is 1.55, we can go here and look, okay, our layer thickness is slightly below 100 nanometers. And this is an example of how basically the measurement is performed then. So red and black are two different wavelengths. We inject our calibration for sensitivity, which is this, and determine this ratio first. Then we inject an antibody, in this case, a specific antibody towards red blood cell EVs. And then we inject red blood cell EVs, and taking the ratio from here, we can then determine the size of this red blood cell EV. We then developed a sensor specifically for looking at red blood cell EVs, because we had a project with Red Cross Blood Service. They had wanted to have a system that they could characterize these red blood cell EVs they're producing, or trying to commercialize now also for research. So we modified our sensor with a thiol. We modified the thiol with protein AG, which is much used for purifying antibodies. And then we block the surface, and then we can inject any antibody of interest that is captured from the FC fragment. And then we can capture the EVs, for example, here. We can have any antibody here or any streptavidin or something else that can capture particles with a specific ligand towards the protein or receptor we have on the surface. then showed that we could use different kind of antibodies against different kind of EVs, for example, platelet EVs, red blood cell EVs, or anti-streptavidin with this surface. So we can see very nice immobilization of these antibodies, and we can use this for capturing particles on the surface. Then we did this validation of the size measurements with liposomes. So we have this sandwich structure with streptavidin immobilized through an antibody on the sensor surface. And then we have bioaccumulated liposomes of different size, 100 and 200 nanometers. We do our calibration, the sensitivity calibration, immobilization of this streptavidin antibody and then streptavidin, and then follow with consecutive injections of increasing concentration of liposomes. And we can see that the black curve is the 100 nanometer liposomes and the blue curve is for 200 nanometer liposomes. They behave slightly different, but that has to do more about the packing, how they pack on the surface, and how the liposomes are actually diffusing to the surface. We can also regenerate this surface and reuse it multiple times. So it's very nice. And here down here, you can see the unspecific binding of the highest concentration. So it's also have rather low unspecific binding of this liposome. Then we did this also for red blood cell EVs, the same thing, sensitivity calibration, red blood cell EV specific antibody immobilization, and then different concentrations of red blood cell EVs. And then the dotted lines here shows the unspecific binding of the same, approximately the same concentrations of EVs here. So by using this capturing technology, we could then determine, by using these two ratios at different wavelengths, the size of the particles, or size of the liposomes, size of the EVs. And then we could compare it to basically two very much used sizing techniques, DLS, dynamic light scattering, and also NTA, which is, nanoparticle tracking analysis. And the upper panel shows now the SPR data, and the average, what we get is the straight lines here, and then we can compare that they actually fit quite well with the data we get from DLS or NTA. Yeah, there is some kind of scattering for the 200 nanometer liposomes, but that has to do more that it's a packing phenomena that 200 nanometer liposome leaves more space between them, and then later on the smaller liposomes are actually somehow filling up the gaps there. But you can see very nicely how they converge, these red blood cell EVs and then 100 nanometer loops liposomes here. Then what we can also utilize this response for is to actually calculate the concentration of liposomes or EVs. And this is a very big question, especially in the extracellular vesicle field, that they want to know, researchers want to know how much of the EVs there are in the sample. And this is also based on real physical parameters. And basically what we utilize is now the initial slope when we inject the particles on the surface. And we can calibrate the form factor of our flow channel with known particles with known size. And here we have used polystyrene particles. And the only unknown we have is actually the concentration of particles. So this is just shown that we get 100 nanometer polystyrene particles, and we get the size that they should show with the SPR signal. And then we can extract this form factor from this information. We can then measure this concentration according to this equation here. You can see the measured concentration with SPR, and this is the concentration determined by nanoparticle tracking analysis for red blood cell EVs. And we can see the red dots, they actually follow very nicely the linear correlation here. For the liposomes, the situation a little bit different. It starts to deviate after when we reach a concentration of 20 micrograms per milliliter. And that has to do that there is a sweet spot of immobilizing particles and not overcrowding the surface to actually get an exact number of the concentrations with this approach. But very nicely, I think we can have shown that we can use this SPR both for size measurements as well as concentration determination of particles or nanoparticles and extracellular vesicles. Then we are very much interested in measuring the protein corona formation of nanoparticles, because that actually determines where your nanoparticles end up in vivo. And oftentimes the researchers use techniques that are very invasive, and they use spinning down of particles and after exposing particles to serum. So basically you can somehow disturb the actual protein corona. So we came up with an idea that we can use this SPR to capture the particles, like I showed before here. Then we can actually inject 100% serum over the particles, flush the serum away, detach the particles, do digestion of the proteins that we have detached from the surface of the sensor, do mass spectroanalysis, and do bioinformatics analysis, and then we can actually identify what proteins we have on the surface of the particles. And at the same time, we get from the SPR data the surface mass density of the particle proteins, and we can calculate kind of protein thickness also on the particles. So this is the model system we used. We had 3 different liposomes, one traditional without pegylated protective layers, one with a protective layer, and then we had, and this F2 also had a fluorescent probe for actually UV-induced drug release in this formulation also, and this had without the fluorescent molecule that do the light-triggered drug release, not UV, but light-triggered. This is the setup. We immobilize the particles on the surface. We can flow plasma or serum over there, and then we flush it, and then we can detach the particles and measure protein quantity. This is how it looks like when we inject plasma, and these different colors shows different particles. You can clearly see different kinetics during the plasma interaction. So the one without PEG, that is actually just a bit of, yes, it has this fluorescent or color compound for light triggering, but it doesn't have PEG. It has a very fast absorber of plasma proteins. And then during injection, it goes down, and during flushing, it flushes away most of the proteins from the surface. Whereas for this pegylated compound, we have slower kinetics during plasma interaction and slightly slower kinetics also in the dissociation phase, flushing period, and it ends up on a higher level than the non-pegylated particle, which is a direct indication of that we have more proteins absorbed actually on the pegylated particles than on the non-pegylated particles, which was a very interesting finding, but it's also supported many studies in literature. So basically what we can then deduce from this SPR data is the soft corona thickness, where we determine the thickness at this point of the SPR data, and the hard corona thickness, where we determine the thickness of the layer and protein layer after flushing. So we can see that the protein soft corona thickness actually increases when we have PEG, and when we don't have the light activated compound in, it absorbs most of the proteins on the surface. and that is the same trend for the hard corona. Normally, one says that you pegulate particles because you want to actually suppress protein absorption. But in this case, this clearly shows that when we have PEG, we have more proteins on the surface than without PEG. So this is a very nice approach then to show which proteins we have on the surface. So we took the next step, detached the particles, and did mass spectrometry analysis and bioinformatics. And there we can then see different this is the blue dots here show depletion compared to plasma, and the red dots shows then over representation compared to plasma. And these could be grouped actually in two separate clusters. So this soft corona for this uncoated particle, and also the pegulated particle without a light activating compound. It's actually the red box here, and we can see that there is depletion in these proteins. Whereas then we have these other hard corona as for all formulations and soft corona for the second formulation. We can see that there are enriched proteins. These proteins are enriched on the corona. And actually, when we do a little deeper analysis of which of these proteins are absorbed on the surface, we can see that the proteins enriched for this formulation 1 here, which is without PEG, is related to immune system activation, which then describes or gives an explanation why it is actually then cleared up from the blood circulation. Whereas the proteins that are absorbed on the formulation F2 and F3, they are associated to lipoprotein binding and enzyme inhibitor activity that correspond to MPS uptake inhibition, which can give us an explanation why they're actually circulating longer. So basically, even though these pegulated particles absorb more proteins, they tend to absorb the right proteins to allow them to circulate longer. So that was more about particle characteristics, and now my talk goes more into how to do measurement with living cells and also on bacterial biofilms. So this is no rocket science in that sense, that you take normal cell culturing approaches, you put your sensor in cell culture, you seed the cells on the sensor, you allow them to grow to a confluent cell monolayer, and then you can start to stimulate the cell and measure the SPR response. This is how it looked like for HeLa cells. So this is a confluent cell layer of HeLa. And if we have a pure sensor, we get this black SPR spectra. Whenever we then coat this with cells, we get this blue SPR spectra. And we can see now clearly that there is a very thick layer here. This TIR angle is changing several degrees, but also this main peak SPR changes in several degrees to the right, which means that we have really thick layer. And if we would compare this to any commercial SPR at the moment, they would only measure in this region from 68 to 70. So it would be completely out of the kind of sensing sensing range in that sense. So that is why this MP-SPR is so efficient for studying these kind of thick layers. So here is just an example, the red line, and when we stimulate the cells with nanoparticles, we can see that the red trace is moving to the right, increasing the refractive index again on the surface. And then when we analyze this as a function of time, we then get the kinetic curve that tells us about the interaction of the nanoparticle with the cell layer. We did a first kind of study together with Jessica Rosenholm from Åbo Academy University in 2015 and published in 2016 about different kind of particles. So we had plain silica particles, negatively charged, positively charged. Then we used polyplexes, which is a complex of negatively charged DNA plasmid DNA with a positively charged polymer that forms spontaneously a nanoparticle in the size of 245 nanometers and very high surface potential. And then we used red blood cell-derived EVs just as an example of natural particles to look on the interaction with cells. So when we measure, then we trigger or stimulate the cells with different charged particles. We can see that the blue trace is the negatively charged particle, the red is the positively charged particle, and we can clearly see a higher response here. And this was also actually then confirmed by confocal microscopy images that there are more of the positively charged particles within the cells than the negatively charged particles. A very nice correlation in that sense. We had an idea during this paper also to actually look on more physicochemical aspects of interacting of nanoparticles with with cells, and that was to measure this interaction as a function of temperature. Because if we do it as a function of temperature, we can then also determine the rate constants of the interaction as a function of temperature. And from that on, we can then calculate the activation energies of the processes. And what we did is that we did these responses at measured the responses at different temperatures. We made a first order reaction kinetic fits to the responses. And then we could then extract the rate constant as a function of temperature. And this is, of course, the very much famous Arrhenius equation, where you can then extract the activation energy of the process. What we found was interesting is that actually we got a kink in this actually plot. Usually it is a straight line as a function of temperature. And this is around 20 degrees, this break here. And actually, when we dig in the literature, we found out that this temperature is actually the phase transition temperature of HeLa cells, cell membrane of HeLa cells. So basically, we found a way also to determine the phase transition temperature of cell membranes if we want to use this as a probe, actually. This was done with plain silica, and then we have coated silica nanoparticles. And we could then extract basically the activation energies above this temperature, the kind of phase transition temperature of the cell membrane, or below here and then above. And these 12 to 18 kilocal per mole basically represent a cell uptake process or endocytosis process, which is Catherine mediated, which we found in the literature also. We did the same kind of studies for these polyplexes, and we made it a little bit more complex here. Also, we also wanted to protect this positively charged polyplex complex with a negatively charged pegylated polymer, so that we could suppress, for example, protein absorption or control the protein absorption, the particles. We didn't do protein corona studies in this, but we wanted to see the effect of this pegylation on these particles. We prepared these polyplexes. They have different setup potential, because when we have this pegylated positively charged particle absorbing the ready-made polyplexes, then it reduces the positive charge of the particle. The interesting thing what we found from transfection efficacy of these particles was that when we increase the amount of this CMD PEG, it actually increased the transfection efficacy also, which is counterintuitive, because if you pegylate something, usually it reduces the interaction with cells, and then you would expect it to actually reduce the transfection efficacy. But here we have an increase. While we increased this, and we wanted to look more into the details of this, so we also made then these kinetic measurements with these two types of particles, positively charged polyplexes, pegylated polyplexes, and you can see, as a matter of fact, there is a lower interaction with these pegylated particles because the signal is lower than for the highly positively charged particle. And when we do kinetics out of this, we can also see that the kinetics is lower for the pegylated particle than the positively charged polyplex. But when we then do these Arrhenius plots at different temperatures, we can see that they look completely different, which actually indicates that they are going through a different endocytosis process. And this is more related to clathrin-mediated endocytosis because the activation energy in that range. But here we see again a change in the energy while we go closer to the physiological temperatures. And we have a much higher activation energy for these particles, which indicates that it's a different type of endocytosis process. Then we do, this temperature is actually 28 degrees, where these transitions start to happen. And speculatively, we discuss this, or I usually speculatively discuss this, so that probably it goes from a kind of a clathrin-mediated endocytosis into more of pinocytosis, which requires more energy because the pinocytosis have to bend and stretch the membrane much more out of the cells. And actually, we can also see preliminary confocal microscope images. We can see that the pegylated particles are actually much closer to the nucleus than here the particles are aggregated and they are much further from the nucleus, which could explain why this transfection is much more efficient for these pegylated particles. Then we just showed as a proof of concept that we can also measure the uptake of extracellular vesicles, and here we use red blood cell EVs to actually look on the interaction with RBC EVs as a function of concentration, and we can see that there is a concentration-dependent increase in the SPR response, and we can also show by confocal microscopy or fluorescence microscopy that actually EVs are inside the cells. We continue this work then by adding platelet-derived EVs to our study, and we change it to PC3 cells, a prostate cancer cell line. And this is just a nanoparticle tracking analysis of the EVs we have from platelets, red blood cells, confirmation that they are actually EVs that we are looking at, and also the protein amounts in the EVs that we have studied. And then the biochemical fingerprint was measured with time-gated Raman also, and just to see that they are actually different in their biochemical composition. The most interesting part from my side was then, or our side with the SPR, was that we measured these SPR responses for platelet-derived EVs, is the black curve here, with red blood cell EVs, is the gray curve. And we can see a clear difference in kinetics. And again, we can move these to first-order kinetic interaction measurement, and we can extract then rate constants out of this process. Then we can use, we injected actually different concentrations of EVs and took the area under the curve here, like we do in pharmacy a lot when we do pharmacokinetics. Then we plotted this area under the curve after 30-minute measurement, and we can see that the platelet-derived EVs actually, according to this, should go in much more in the PC3 cells than the red blood cell EVs that seems to actually level out at some point. This was actually then also confirmed by confocal microscopy, where they could show for much longer time points than we are doing for half an hour here. But for longer time points, there is also the same clear trend that platelets are going more readily into the cells, whereas the red blood cells are actually leveling out. And the interesting point here from the SPR point of view is that if you look on the relative ratio here at 3 hours and half an hour, it is exactly the same what we determine with SPR, kind of then confirming that we are measuring the uptake process of the EVs with SPR. And my last example relates to bacterial biofilm growth and, and actually interactions with nanoparticles. This is actually manuscript got accepted on Monday this week, so it's very fresh out, not yet in print, but hopefully soon. So basically this shows the process of how a biofilm grows, and the main motivation for this study is, of course, because we have this antimicrobial resistance is a very big concern in the world. That I think WHO has put it as one of the top 10 threats for human health in the world, and it's very difficult to study kind of the interactions with drugs and nanoparticles with biofilms, and maybe also understand the growth mechanisms of biofilms. So we thought we could actually monitor the biofilm growth in real time, first of all, to characterize how biofilms are growing as a function of time. And then we can use that biofilm later on to inject some antimicrobial compounds and then see how it reacts to this antimicrobial compound. And here we actually utilize another parameter. Now, so far I have been showing all the results is this SPR peak minimum change. When it changes to the right, we have an increase of refractive index. When it goes to the left, there is a decrease. But now we can also utilize this intensity of this peak. That tells us more about how even the layer is on our sensor surface. So if you have a very much island-like structure, as biofilms usually grow from single bacteria to colonies, islands-like, and then when the matrix start to grow, then it becomes a homogeneous film. So we had the idea that we can monitor also the intensity changes here, which also reflects the stages of the biofilm growth. What we then did, we measured the change in the peak position, which gives us this nice curve. We were measuring continuously for 24 hours while we were just making a bolus dose of bacteria, and after that we just circulated or pushed in growth media or nutrition media to the bacteria, and they start to replicate and grow and then form a biofilm. When we then use this intensity data, we saw actually that, okay, we have a delay time here, but at a certain point, this intensity starts to grow. And exactly at the point where we have steepest slope here, it then turns around and then it goes down and comes almost back to the zero level. And this is now an indication that we have first a growth of colonies that scatters the light from the surface of our sensor. And when the colonies start to produce, for example, the extracellular matrix or material in the biofilm, they become more homogeneous films. And when it becomes a complete biofilm, then we get a total reduction of the intensity change. So we can pinpoint the exact time point of the process of biofilm growth. We did also optical modeling on this, and to determine that the biofilms we were using in this study was from 2.5 to 3.3 micrometer thick. But the next step was then to look on differently charged nanoparticles, how they behave and interact with this biofilm. And this was based on a cesium oxide core, which was coated with a mesoporous silica coating. And then the mesoporous silica coating was then modified with polyethylene imine or amines to get positively charged particles. That was further functionalized with succinic anhydride to get negatively charged particles, as well as then quite neutral particles. And here we can see the sizes of the particles around 300 nanometers, and the surface or setup potential, you can see highly positively charged for the P particles, highly negatively charged for the succinic anhydride particles, and then, well, not really neutral, but closer to zero for the for the acid anhydride particles. And when we then injected these particles, we could see a clear difference in the responses. Now the first part here, you can see before the green vertical line is the biofilm part I was showing in the previous slide. After that, we inject the particles, and we can follow the particle or the change in SPR angle as well as the change in SPR intensity. And we can see clear difference for the positively charged particles compared to the negatively charged and neutral particles. So there are not much interaction with this negatively and neutral charged particle, whereas in the positively charged particles, really a tremendous increase in the signal. And this was also confirmed by confocal microscopy, that we could actually see that the positively charged particle penetrated through the whole biofilm all the way to the bottom, whereas the negatively charged and neutral, they did some penetration to the middle part, but not all the way down to the bottom part of the biofilm. And this is, of course, very interesting because we could then utilize, for example, these positively charged particles to deliver drugs all over the biofilm and not only to certain part of the biofilm. And this is, of course, one of the big problems with antimicrobial drugs, that they cannot penetrate all the way into biofilms. And then we can also use these two parameters to look a little bit about the mode of action of what is happening. So this upper line here is now the growth path of the biofilm itself. So for each measurement, we did some averaging of 2 to 3 responses, and we can see that the trends are the same for all the biofilms. So the mechanism of growing is the same. But if we would compare different biofilms, we could from this plot maybe deduce different type of growth mechanisms later on. And the bottom part shows now then the particles. And we can see for the positively charged particle, it's completely different behavior than for this other. And in the future, it would be nice to look more into more particles to see if we could kind of somehow be able to predict the mechanism of action of these particles with biofilm. Actually, that was all. These are the funders that have been helping out our group for many years, for the past, I would say 12, 13 years altogether. And I'm happy to get any questions or comments.